Your liver was never designed to work around the clock, yet constant eating can slowly overload it with fat, disrupting how your cells manage and utilize energy efficiently even if you rarely drink alcohol. But hidden inside your biology is a powerful survival system designed to support normal cellular maintenance and metabolic recovery processes. Every meal places your liver into an intense processing cycle. As carbohydrates are digested, glucose rapidly enters the bloodstream, signaling insulin to move that excess energy out of circulation as quickly as possible. The liver responds by storing part of that glucose as glycogen, a compact backup fuel source the body can access later between meals or during physical activity. In many ways, glycogen functions like a rechargeable biological battery designed for short-term energy stability. The problem is that this storage system has limits. The liver can only hold a relatively small amount of glycogen before its reserves become saturated. Once that threshold is reached, the body must find another place for the remaining energy. Instead of storing more glycogen, the liver begins converting excess glucose into fat through a process known as de novo lipogenesis. When frequent eating keeps refilling the system without enough recovery time, fat may gradually begin accumulating inside liver tissue over time. Modern eating patterns rarely allow that glycogen battery to fully empty. Many people wake up, snack constantly, drink calories throughout the day, and eat again late at night. From the liver's perspective, the storage signal never truly stops. Over time, excess energy begins spilling beyond normal glycogen reserves and gets converted into fat, gradually accumulating deep inside liver tissue. Biologically, this creates a state where the liver spends far more time storing energy than accessing it. The system becomes trapped in a near-constant cycle of intake, storage, and overflow, leaving very little opportunity for meaningful metabolic recovery between meals. Several hours after your last meal, the body finally begins moving out of constant storage mode. As digestion slows and blood sugar stabilizes, insulin gradually falls instead of remaining constantly elevated. This hormonal change allows another powerful signal, glucagon, to take control. While insulin is designed to store energy, glucagon does the opposite. It tells the body that it is time to start accessing stored fuel rather than continuously packing more away. Inside the liver, glycogen reserves that were filled after previous meals can now begin supplying energy back into the bloodstream. Under glucagon's guidance, the liver slowly breaks glycogen back down into glucose and releases it in a controlled, steady manner. This process helps maintain stable blood sugar even while no new food is entering the body. More importantly, it gives the liver a temporary break from non-stop storage mode. For the first time in hours, the body begins moving away from accumulation and toward metabolic recovery instead. In some individuals, several hours into a fast, the body begins approaching the limits of its readily available glycogen reserves. With little incoming fuel left to rely on, the liver must transition into a different metabolic strategy to keep the brain and muscles supplied with energy. This marks a major shift in human metabolism as the body gradually moves away from primarily burning glucose and starts relying more heavily on stored fat for fuel. During this phase, fatty acids begin leaving storage tissue and traveling to the liver, where they are converted into compounds called ketones. Ketones provide a stable energy source that many organs, including the brain and heart, can efficiently utilize during periods without food. Energy availability also becomes far more stable throughout the day. Rather than depending entirely on recently consumed calories, the body starts drawing more energy from its own internal reserves, including fat stored around the liver and abdominal organs. Ketone production is not the only metabolic change taking place during this stage of fasting. Another powerful survival hormone, growth hormone, also begins rising as the body adapts to lower incoming energy. One of its most important roles is helping protect lean muscle tissue, while stored fat becomes a larger fuel source. From an evolutionary standpoint, this mechanism helped humans maintain strength and mobility during temporary periods without regular food intake. Maintaining this balance is extremely important for long-term metabolic health. Rapid weight loss methods can sometimes force the body to break down muscle alongside fat, slowing metabolism and weakening physical resilience over time. A properly regulated fasting state creates a very different hormonal environment. Instead of aggressively sacrificing muscle for energy, the body becomes more efficient at mobilizing stored fat reserves. This allows the liver to gradually reduce part of its accumulated fat burden, while overall energy, stability, and metabolic function remain better supported throughout the process. As fasting continues, the body gradually shifts more attention away from digestion and toward internal repair. Around 12 to 18 hours after eating, many cells begin activating a remarkable biological process called autophagy. The word literally means self-eating, yet the process functions more like a highly organized cellular recycling program than destruction. Its purpose is to help recycle certain damaged or dysfunctional cellular components as part of normal cellular maintenance. Damaged proteins, worn-out cell structures, and leftover metabolic debris can slowly build up inside tissues after years of continuous stress and nutrient overload. During autophagy, cells collect much of this material and transport it into specialized recycling compartments where it can be broken down efficiently. Some of those recycled components may later be reused to support newer cellular structures or provide additional energy during the fasted state. This temporary shift toward cleanup and repair may support healthier metabolic function and cellular resilience throughout the liver and other organs. Beyond general cellular recycling, the liver also activates a far more targeted cleanup mechanism during extended fasting known as lipophagy. Unlike standard autophagy, this specialized process is associated with the breakdown and recycling of certain fat droplets stored inside liver cells. Years of chronic overeating, elevated insulin, and constant snacking can gradually overload the liver with internal fat deposits that interfere with normal metabolic balance. Lipophagy allows part of that trapped fat to be mobilized and converted into usable fuel instead. What makes lipophagy especially fascinating is that the body already possesses this built-in mechanism for managing excess internal fat when the hormonal environment finally allows it. No detox teas, extreme cleanses, or artificial products are required for the process to occur. These processes are being studied for their potential role in supporting normal metabolic function during periods of fasting. After the liver begins reducing part of its accumulated fat burden, many people notice meaningful changes in both physical energy and mental clarity. Some people may experience steadier energy and fewer noticeable blood sugar fluctuations, and the body grows more comfortable relying on a steadier mix of stored fuel sources between meals. More energy now comes from ketones, which can serve as a stable alternative fuel source for organs such as the brain, heart, and muscles during fasting states. Many individuals describe this phase as a noticeable shift away from the constant cycle of cravings, energy dips, and afternoon fatigue that often accompanies unstable eating patterns. Hunger signals may begin feeling calmer and less urgent as the body adapts to accessing stored energy more efficiently. Some people also report feeling mentally clearer during this transition. Instead of depending heavily on repeated glucose spikes for temporary bursts of energy, the body may gradually become more metabolically flexible over time. By the later stages of an extended fast, the liver may shift more of its activity toward maintenance and metabolic recovery processes instead of continuous digestion. Somewhere between 18 and 24 hours without food, markers associated with metabolic stress may gradually improve in some individuals as the organ spends less time processing incoming nutrients. This shift creates a calmer internal environment that can support normal cellular maintenance and recovery more effectively. Two important enzymes often associated with liver stress are ALT and AST. When liver cells become irritated or damaged, elevated levels of these enzymes can sometimes appear in the bloodstream as signs of metabolic strain. During periods of improved metabolic regulation and reduced fat accumulation, those stress signals may begin stabilizing over time. One remarkable feature of the liver is its regenerative capacity compared to many other organs in the human body. Under healthier conditions, the liver has a notable ability to regenerate and maintain normal function under supportive conditions. The period immediately after a fast can strongly influence how the body responds metabolically in the hours that follow. Once eating resumes, insulin naturally begins rising again to help move incoming nutrients into storage and restore depleted glycogen reserves inside the liver and muscles. This response is completely normal and plays an essential role in maintaining energy balance after a fasting window. Food quality, however, still matters during this transition phase. Highly processed meals loaded with refined sugars and excess calories can quickly overwhelm the system and encourage renewed fat accumulation inside the liver. A slower and more balanced reintroduction of food is often easier on digestion and metabolic regulation. Meals centered around protein, vegetables, fiber, and healthy fats generally create a steadier glucose response while helping support satiety and energy stability. The goal is not perfection or extreme restriction. The liver does not need punishment to recover. In many cases, it simply needs consistent periods where the body is no longer trapped in non-stop storage mode. This content is for educational purposes only and is not medical advice. If you found this video helpful, please like and subscribe to support the channel. Thanks for the support.